Hey, welcome. This is Dr. Michael Timms, and this is going to be a lecture on quality in the production of herbal medicine. And we're going to focus on three areas: distinct contaminants, issues of botany and the effects of botany on both identification, but also on phytochemical makeup, and then adulteration. So some of the examples that we're going to go over in the contaminants area, we're going to really look specifically at some mold and bacteria. Look at some methodology so you can get an idea of how this might happen in a workplace. We'll review some echinacea botany, some elements that affect it based on the movement of gene flow and how that affects the actual efficacy of the plant. And then adulteration, we'll go over some of the issues that are currently in the news around adulteration. What you'll get is a lot of new vocabulary. And it's always good to look at the words that you don't know. But really it's about the ideas. Do you understand the main ideas? And the fact is, quality is a really complex set of those ideas. It's not simple. You're not going to learn it in one take. So let's jump in. So the contaminants are coming from multiple places. You have a plant that's in the soil and in the air is being exposed to fungi, spores, bacteria, viruses, and you go through processing, and if you have the right conditions, all of these can grow and grow to a point where they're beyond what's considered an acceptable or safe area. So in mold, I think the big thing is mold isn't everywhere. It's not homogeneous in its distribution. So you tend to have pockets of contaminants, either when you have a cut and sift, when you have whole plants that are dried, when you have powdered materials. So the important thing is, if you're going to go and test, if you're in the lab for a company or your own, that you do micro-testing, that you have to figure out some statistically significant sampling procedures in order to make sure that you try to address all the different potential pockets. That doesn't mean you go everywhere, but you have a randomized sampling and you get enough samples that you can account for it. And then the other thing too is when you do this, there are matrix interference or product inhibition elements. That is, some of these medicinals actually have antifungal, antibacterial properties. It doesn't mean that the fungi or the bacteria aren't present, but the plant is actually interfering in terms of their growth. And we need to grow them, as you can see on these plates, in order to identify them. So on the left is Fusarium equiseta, which is grown on potato dextrous agar, which is a fairly normal general media for growing up fungi. And you can see the filaments grow both horizontally, but also aerially up towards you, away from the bottom of the plate. And it's already begun to sporulate. You see some spores forming. In this right-hand picture, what you're seeing is a mixed culture. You're seeing some, what looks to be some Fusarium, some penicillin, and not clear exactly what that could be, a number of things. So this is really a pure culture on your left, and you have to do some work to get it there. This is normally what you'd see if you took some dried plant material or powdered plant material and sprinkled it on an agar plate with the appropriate mixture so the fungi or bacteria can grow. This is what you tend to see. So some of the high-risk contaminants, these are the most common molds, both by their presence in the soil, but also their potential to have an impact on humans. So Alternaria, Aspergillus, Chlorosporium, Fusarium, Penicillium, and Stachybotrys. And the mold in itself can be allergenic, but the element that really makes it more difficult is the mycotoxins that they produce. And we'll come back to that in a minute. So generally, the picture on your left is something that you see in a microbiological lab, where you have biosafety hoods, you have autoclaves for making the PDA or the agar plates, incubators for the growth at an optimum temperature. But at some smaller firms, you can adapt. So you can set up a sterile work area by not only wiping down surfaces with 70% ethanol or methanol, but having an actual Bunsen burner that creates through convection current a really nice sterile area here. So you can actually work out in the open if done right. The additional elements would then be to have some kits. So let's go over some standard methods for testing, and then I'll show you some kits that can be very useful in a small workplace. So cultural methods, we talked about that already. This is where you take agar, which is a sort of a solid media that's squishy, and you add general or selective or differential nutrients. That is either nutrients that all fungi can grow on, or selective, meaning that you are selecting for something that only grows in this environment. Or if you want to differentiate, you would add elements that suppress the growth of certain microbes while encouraging the growth of others. And generally when we use those plates, we're talking about plating techniques. How do you actually go about putting whatever you want to have measured on the plate? So you can pour it on, you can spread it, you can streak it. Any of you that have had microbiology will remember some of these. And then you'll use things like most probable number techniques, membrane filtration, dilutions and enrichments. Right? This is where you're trying to actually come up with a colony forming unit, and that's where you dilute your sample enough so that you can get a plate that grows little round circles or colonies or whatever you're trying to measure, and it needs to be around 40 colonies or less so that you can count them, and then you come up with a colony forming unit, and that's acceptable. And you can also do biochemical testing. So I don't know if you remember your micro labs, but each of these compartments is a special media, and it tends to be selective media. Only certain types of microbes will grow in there. And you pull this pin out all the way, and you inoculate the tip and push it back through. And you do it in such a way that you're simply inoculating. You're putting whatever microbe you're looking at in each of these compartments. And the color reaction then will give you an indicator of what type of, in this case, bacteria is present. So here's some things that you can do. The 3M sells a number of very good plates, count plates. They are literally small, thin applications of agar onto a backboard with a cover. You can work in the open setting, as we mentioned earlier, with these types. You can inoculate them very easily, and you can get very specific type of plates. So you look at coliform or mold or bacteria, or you can look at yeasts. So this is increasingly becoming the way of doing things, because you don't have to have all the prep lab space for it. On your right is a agar plate where you have agar that's already been poured in and is solid, and you plate it, incubate it, and you count. In the other, it's a pour method where you inoculate it first. You pour in your warm agar, not hot, because you don't want to kill your microbes. You then allow it to incubate, and you begin to see subsurface colonies as well as surface colonies. And they're used for different types of microbes. And these are what some plates look like. So this is going to be a penicillin on your right, on your left. On your right are bacterial. Oftentimes bacterial colonies are going to be shiny, certainly shinier than most fungi. And here's some mixed colonies where you get a differential plate here. This is a general plate, and you can see different types of colonies growing on them. This is normally what you'd pick up from your plant material. And here's some specific fungi and what they look like, both in terms of their growth on a plate, and these are the spores which you would look up under a microscope and would help you identify them. So the combination of these two allows you to identify which fungi you're looking at. And here's Aspergillus. Notice the green color, but the spores forming in the middle of the colonies. Here are these long spore sequences. This is Cladosporium. This growth habit is very indicative of it. And this is Fusarium. In this case, you've got the spores, and you've got some of the spores actually breaking dormancy, and you're getting mycelia growing. And this is Penicillium. Again, signature microscopic image and combination with how it grows. And this is Stachybotrys. So the real risk of these, I mentioned earlier, are the mycotoxins. And this link is to the place on NCBI, which is the PubMed and National Library of Medicine at NIH. And these are the mycotoxins that can really cause some issues for human beings. And that's what we're looking for, because you can have mold and you can then dry your material out and kill off the mold. But if it's already produced the mycotoxin, that will be present, and you won't be able to tell based on morphological features. You won't be able to spot the mold. So that's why we really want to learn how to process things effectively so that you can eliminate some of this. And here's really a look at a number of different organizations that set standards for the herbal product arena: APA, NSF, United, U.S. Pharmacopoeia, World Health Organization. APA is American Herbal Products Association. And you can see this is for dried, unprocessed, and powdered botanical extracts. And these are the limits, actually. Microbial counts 10 to the 7th. These are numbers, and you can see that they differ. And they certainly differ when we begin to go to extracts as opposed to dried, unprocessed. And this can be found on APA's site. But these are some parameters that seem to be acceptable to the FDA and are good numbers to look at if you're either doing this work yourself and if you're using an external lab. Now, one thing I would encourage you to do is the quality assurance course. Actually gets you this hands-on experience looking at the results of your work, actually doing plate counts. And it's a great way to take the theory and put it into what would look like in a production setting. So that's a quick rundown on contaminants. Now what we'll go to is we'll look at echinacea as an example of this, because really it's a very popular herb, and there's some interesting characteristics about it on a botanical level that influences the kind of medicine that we end up getting from it. And this is really on how we define species. So echinacea's got a very rich set of phytochemical constituents. You've got the phenylpropanoid derivatives, chicoraic acids, chelicosides. You've got the arabinogalactan proteins. You've got the alkylamides, which are lipophilic compounds, and there's a whole set of them. And what's interesting is we also look at it from the point of view of at least 3 major species in trade. And you can see the augustifolia at the top, Pallida in the middle, Purpurea at the bottom. And we also look at it from the point of view of using the aerial portion, stems, leaves, flowers versus roots. So it's a complex medicine in many respects. So when we look at the phytochemical makeup, we also then have to deal with how we make the medicine. So the alkylamides and phenolics tend to be more concentrated in the roots. They're better being extracted in an ethanol extract, less hydrophilic. It is hydrophobic in nature. And these compounds tend to block inflammatory response. They tend to help in the suppression of infection symptoms. The polysaccharides and the arabinogalactans are more concentrated in the aerial portions. They are hydrophilic, thus the water extract, and they tend to enhance immunity via increased cytokine presence. Now, some other elements are that when we dry, at least in the Pallida we have this evidence, and it wouldn't be difficult to assume that this occurs in the other plants as well, are artifacts that are formed. That is, they're particularly in the alkylamides and some of the chicoraic acids, you have compounds that get changed in the drying process. And Out of Europe, you see a lot of fresh juice extracts because they believe that you got more of the medicinal effect based on that. And this is something that I think really needs to be standardized a little bit better. What type of medicine you're getting from dried versus fresh and the type of extract. And this gives you a picture of the three species. And if we look just at the root tissue and we look at the phytopropanoid derivatives like cynarin, echinocoside, chicoric acid, and the echinocoside derivative, and the verbascoside, and then 4 different types of alkylamides. What you see on the right are HPLC chromatograms, which measure the presence of those individual compounds. So you can just take a quick look and see that the distribution, even if they were all present, all compounds were present, the distribution is very different. We see in the first one more of 3 and 5, in the second one more of 2 and 4, and finally in the last one, particularly more of 7, 8. We can see that 1 and 2 are missing from Purpurea. From Pallida, we can see that 5 is missing, and not any of 1 apparently. And then in the bottom one for Angustifolia, you don't see any of 3 or 5. And so you could ostensibly look at something like this if we got enough samples and ran this enough times and used some statistics to define what the level of variation would be from product to product. We might be able to actually use this as a way to identify what species we have present. Only if they were alone. Now, if they got mixed together, we wouldn't know. But we tend to get a lot of variation. So we look at the alkylamides, and we see that they're most abundant in the roots of Purpurea and Angustifolia, and much lower in the aerial portions, and much lower in Pallida in general. And we can see that based on these numbers. If we look at the 6, 7, 8, 9 here, we do see some in Pallida, much more in Angustifolia, and to some extent Purpurea. So this is the alkylamides that we've found and identified and named, and there are others that have not been named yet. But this is a very similar structures. They're all linear. These are cannabinoid receptors. For some of you, you've already begun to investigate that element. And here's a study where they took, from commercial products, they analyzed the alkylamides. You can see these 1, 2, 3 are all liquid tinctures. The first 2 are pretty much the same, Purpurea flower and root. The third one is Purpurea and Angustifolia roots. In 4, 5, and 6, or actually 4 and 5, you have dried capsules. In one, both Purpurea. In one, you have aerial parts and then roots. And then the 6 and 7, 8, and 9, what you have really are the same Purpurea aerial parts and Angustifolia roots. And what we're doing is we're comparing 2 different products. So this is the distribution of the alkylamides. So remember, these first 2 were just Purpurea, and the addition of Angustifolia was in this third one. And you can see that there are some differences here, 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 where you have compounds that were not present. But since it was a mixture, right, remember it was Purpurea and Angustifolia, this is unexpected, the absence of that there. So part of this speaks to the ability to recognize what compounds to expect, but also says there's natural variation, which makes it very difficult. Here you have a combination of comparing the aerial parts versus the roots. This gives you a really nice, clear picture of how the roots have much more of the alkylamide fraction. And then here we're looking at pairs of the same products, 6 and 7, and then 8 and 9. And what this really speaks to is the natural variation either in the processing and the medicine making, or just in the biological material itself. You can see that this alkylamide is missing from 7. Here you can see where this acrylamide is missing from 9, and this acrylamide as well. So does it make any difference? Would those two products have the same effect? We haven't really done enough investigation, but you might in a practice, in your clinical practice, or you might in just some trials that you might do as a manufacturer, might look at some of those issues in terms of comparing how much effort you have to do to make a high quality, efficacious product versus something that maybe isn't as much work. And this goes back to the old Galen's galenic extract. What's good enough? Can you just do a 60% alcohol extract and that's enough? Or do you need to do more specialized extractions, doing both water and hydrophobic extractions and then combining them? This is the picture of Echinacea purpurea, and we're looking at the aerial portions, the flower and the stem, the leaves, and then the roots and rhizome. And the reason is, when we look at the variation by tissue, and these are different samples, 1, 2, 3, 6, 7, 8, 9. What we're seeing is the mean alkylamide levels in milligrams per gram dry weight, followed by their standard deviation, and the bold numbers are significantly different. So what we're really saying is, by standard deviation, is this number is really could be anywhere from 0.6 to 0.2. This is a plus or minus in here. So here we're seeing in these two samples of Echinacea purpurea, they have similar amounts, and the highest are in the stems. In these three, the highest amounts are in the roots. And then finally, in the combination of 8 plus 9, we're back to the stems, and much, much higher amounts than we would if we'd added these two together. So again, this speaks to some of the uncertainty of these phytochemicals and their composition and what role they play. So if we get a product like the 8 and 9 combined, and we're getting this much alkylamide fraction, and we run a clinical trial with that versus something like 1 and 2, will the differences be that much greater? Will one trial say it's effective and another say it's not? And so when we see these conflicting results coming out of clinical trials that are being reported on PubMed, that's one of the things we have to begin to think about, is we're not providing a standardized dose of a pharmaceutical agent, single entity. We have a biological product with tremendous variation, and there are lots of things to think about. So another one of the fractions, we were just looking at acrylamides, is chicoric acid, which is a phenylpropanoid derivative. Now this tends to degrade during processing and juice extraction. Chicoric acid is in a lot of different plants. We see it in black tea. Black tea is a fermented green tea leaf, and it's dried and processed. Just a simple addition of some ascorbic acid or alcohol for anything greater than 45% can actually inhibit the degradation. So if this is a compound that is fairly water soluble, it helps to have at least 45% ethanol in your extraction to keep it from being degraded. And that degradation process can occur because there are enzymes in the plant itself that, once that material surrounding the cell membranes get broken down, when those enzymes had been sort of set aside and stored in another area of the plant cell away from the chicoric acid, now they're allowed to mix freely, and they begin to literally degrade the structure of that compound. So that slight addition of ascorbic acid or that ethanol helps with long-term and short-term storage during extraction and then on the shelf. So here's some elements that we need to think about when we start to think about quality are, what could these enzymes actually be doing? Now you're going to see this in your work with the elderberry, that there are enzymes in elderberry that can also break down, in the case of elderberry, the anthocyan and glycosides. So the other issue that really comes into play on the biological side is something called hybridization. So generally when we speak of a species, we say it can only reproduce amongst itself. But the echinacea taxonomy is a little bit confusing. Based on morphology, there have been 7 species with 2 varieties. So you've got the species of Atrorubens, Labigata, Pallida, Purpurea, Paradoxa, Simulata, Sanguinea, Augustifolia. But we know that things happen on a genetic level where you actually have plants that hybridize, so oaks and hawthorns and birches and bugleweeds and thymes that begin to, if they're in proximity with each other, they actually can cross or mate, and you begin to get gene flow altering the phytochemical signatures. So Echinacea pallida and Augustifolia are indistinguishable without their flowers, morphologically speaking. They grow in the same area. They will cross with each other, and you'll get genes moving back and forth. And so therefore you'll get a hybridized plant, which is a mixture of the two. Well, when it comes to ensuring, we've already seen that the phytochemical distribution can be different. So how do we account for that then when we begin to make medicine? Binns was a researcher out of Canada, Shannon Binns, who did some mathematical analysis, different from the morphological analysis. And what she found was that there are actually 4 species. So there's the Echinacea subgenre Echinacea. So you have genus, subgenre, species. And so for her, the 4 species are Purpurea, Atrorubens, Labigata, Pallida, and that there are varietals associated with them. And one way to take a look at this was to say, all right, well, let's look at the phytochemical profile, the metabolic profile. And they looked at the lipophilic metabolites. So these would have been largely the acrylamides, but also some other compounds. They looked at 40 accessions being collections, and the accessions need to ideally come from different distribution areas, so you're not looking at the same gene stock or environmental influences. And they took 3 samples of each, and they measured all these metabolites, and they used some statistics, weighted PCA and clustering, which is a way of making sense of the distribution of those compounds in all these different plants. And they used a very special math to compare the mathematical model that Binns had versus the morphological model that McGregor had started with. So you had McGregor, which is morphological features, and a system more recently developed by Binns. And these were the differences based on this Canberra distance, where we see in panel A the 7 species and in panel B the 4 species. And this is what they found. Generally, if we just look at the lipophilic metabolite profile, it's the distribution of morphological features, the 7 species, that shows up in the way these lipophilic compounds are distributed. And what it says is that these alkylamides don't necessarily evolve in a convergent manner. That is, to converge, meaning a bunch of similar plants all sort of come to one endpoint in a response to a particular geographic or environmental condition. So it speaks to the fact that these plants literally re-emerge with different genomes depending on how they hybridize, how they mix their genes with other very similar species. But what it does allow us to do is to say, okay, the morphology is a good way to define at least the acrylamides in a consistent method. And we can use a typical fingerprint profile, which can then be validated by looking at more populations to sort of say, okay, this distribution of alkylamides looks like Pallida or Atrorubens or Augustifolia. So here's where we get some agreement between the phytochemical ratios and makeup and a species name. So finally, we're going to get to adulteration. Interesting thing in at least the U.S. market is herbal products have always been adulterated. We've had snake oil cons from the beginning of the game, right? They literally named them love potions and snake oil to help keep snakes away and to prevent the toxicity of the bites. And in the current marketplace, the FDA holds manufacturers legally responsible for preventing tainting of products in the U.S. It's a safety concern for them. And some of the more recent examples are the presence of synthetic drugs added to sexual enhancement, weight loss, and bodybuilding products. This is a market, I'm sure you know, that is ripe for manipulation because these products sell huge amounts, and people won't necessarily think through these elements. But we also have things like black cohosh, which in the U.S., Actia racemosa is an excellent herbal, but you can get Asian species, which can be slightly toxic. So we can see some adulteration in that marketplace based on the Asian species. And this is what it might look like. This is Actia racemosa, what we use in our marketplace. And these, again, is an HPLC chromatogram. And this is a distribution of individual phytochemicals. You can see there's a big difference between all four of these. The bottom three are Asian varietals, or species. And you get a very different distribution of phytochemicals, either absence of, or greater or lesser amounts of, or the presence of compounds that aren't in the racemosa. This is a series of black cohosh products all the way down. And we can see these are sort of expected portions of the chromatogram. We would expect that in racemosa. These are not. So if we just go down, you can see that some of these contain elements of an unexpected constituent or chemical, along with some of the expected. Now this could be a mixing of an adulterant with racemosa. In this case, there's none of the expected amount. You can see that some of these have much greater amounts than others. So maybe you're getting not as much as the label claims. So this is a methodology that allows us really to go in and identify potential adulterants. Now, when we ground up material and we can no longer identify them based on morphological features, we can look at some of the genetic identity. But the genetic identification of extracted botanicals is really a big problem. So we had a report out, remember, from the Attorney General of New York, and it brought up a bunch of concerns around adulteration and the use of genetics to identify them. But when we extract, we tend to target a broad group of compounds based on whether they dissolve in a solvent. And there's a tendency to enrich the components that we're actually interested in and removing those that we're not interested in. And the most comprehensive approach to botanical authentication is based on these chemical compositions. You can see that we've done that for a number of plants already. The black cohosh, the echinacea, and then using some powerful statistical models. So this spectral fingerprint, what we saw in the HPLC, allows for the discrimination between species, oftentimes growing locations, conditions, even processing, where we can see where heat or light has had an effect on compounds that are expected to be broken down based on exposure. So the New York Attorney General is using this adulteration as a platform to higher office. Really, this is someone that wants to potentially become the governor of New York. And in the process, has brought a bunch of things to light. One of them was a devil's claw, where Herbal procumbens is listed in the Herbs of Commerce #1, first edition. But it's been used along with the Zahiri interchangeably for a long, long time. This is traditional. It's not an attempt to adulterate. But based on the Herbs of Commerce first edition being the actual signature plant, anything else is then considered an adulterant. So what's happening is AHPA, American Herbal Products Association, is revisiting the third edition and including the Zahiri in there. And that then will be accepted by the FDA as the indicator of what species should be found in trade. So even though this isn't really an adulteration, it speaks to other issues such as making sure whatever legal document is the source of what should be found in a product, that is devil's claw. It notes that there, in some cases, as in the echinacea, you can use multiple species and still have an effective medicine. The other thing that was brought up was the DNA barcoding. And barcoding is really useful to identify original plant material with missing taxonomic characteristics, but not finished products. Some of the things that happen, though, is when you look at genetics, in barcoding, you're measuring, trying to find a region of plants that you could measure in all plants and differentiate them. We've been able to do that with animals, but not with plants. So a primer is simply a part of the genome that surrounds a gene of interest. If you can find the primer, then when you put in the appropriate ingredients to replicate or make more DNA, it will find that primer and make the DNA that follows into a copy. The reason for doing that is you want to amplify the amount of DNA that you're measuring so that you can see it. But what ends up happening is when we process herbal medicine, and particularly when we do extracts of them, right, we're actually trying to pull out the phytochemicals, we degrade the DNA so that very little is left. Often you'll have multiple plant species with the identical sequence in that part of the DNA that they're trying to use for the ability to differentiate the barcoding area. We are using those primers to amplify the amount of DNA so we can see it, which means we're also amplifying non-specific DNA or contaminant sequences. And the other part of it is in DNA, we can't identify the plant part or whether the appropriate growing conditions were used or if it has the appropriate chemical profile. So DNA is a limited tool. Now, when barcoding was used, remember it's a process, not necessarily technology, it's only able to authenticate about 10% of crude raw materials, none of the finished products that were made with extracted ingredients. But when we went to specific DNA that could be used for that plant, fit for purpose, 90 to 60% could actually be authenticated, meaning identified and shown to be a particular species. This gives you a sense of using a single set of genetic markers here in this column, and that was the accuracy. And then when multiple gene markers were used, the accuracy went way up. So this is becoming, instead of having one DNA barcoding method for all plants, what it turns out we have to do is we have to find a method that's specific to a plant to measure its presence. But now, that doesn't necessarily tell you what else is there. It just tells you that that plant is present. So the other thing we have is that most of the DNA is run in labs and it's proprietary, and we can't reproduce it, so we can't test it. And the other thing that DNA has a hard time doing is it can't differentiate between the herb tested in either inorganic material that's been added or foreign organic material. So let's just take a look at that. Here you have dicalcium phosphate, which you often find as a filler. So it was added to a small percentage of E. purpurea leaf, powdered, and the DNA results said that it was 100% purpurea because it can't differentiate the presence of the dicalcium phosphate. Now, 400 milligrams of inert filler and 100 milligrams of a particular botanical. Now, this would not have passed the appropriate macroscopic, microscopic, or even standard chemical tests. So here is this high-end DNA method that really can't do as much as these simpler approaches. We already know the idea that if you have non-target DNA, because you're always going to have some material there, as long as it falls below the accepted level, that's fine. But remember, when we do DNA testing, we amplify that amount. So we can actually change the sensitivity. And what might show up is that you have huge amounts of a fungal contaminant in your product, when really you don't. You don't have a good measure of concentration. So finally, in the future for this type of approach, plants, there are thousands of them, species. And it requires time to refine methodology, build authenticated reference material, so that we can go back and say, this is what DNA from this species looks like. It has to be fit for purpose. And really the focus now is looking at chloroplast DNA. It turns out chloroplast is where the chlorophyll is present. This is where the photosynthetic machinery of the plant is. And chloroplasts are everywhere. They're in root tissue. They're just in lower copy numbers. So in a leaf, you're going to have huge numbers of chloroplast DNA, and they tend to be these simple circular portions of DNA. And this is where the real work is being done, is to say, all right, let's see if we can't differentiate species based on the DNA in the chloroplast and see if there aren't some differences. And part of that has to do, because this tends to be more maternally inherited by the plant versus nuclear DNA, which works like any sexual reproduction. It's a mixture of both the parents' DNA, and therefore much more heterogeneous. And in this case, chloroplasts may in fact have some preserved elements that are unique to a species. So that's really the sort of the future of the work for DNA and identification in terms of looking at adulterants. So I hope I've been able to go over new methods that you're going to see in the press and the strengths and weaknesses compared to contemporary approaches. Finally, this is a great program. This is American Botanical Council, which puts out Herbalgram, American Herbal Pharmacopoeia, which puts out a series of monographs, and University of Mississippi's National Center for Natural Products Research. I recently had an adulterants paper published for them looking at Golden Seal, because I had done enough of work on it. And Golden Seal had both historical and current issues in terms of adulteration. If we look at the pricing in that graph on the bottom right, you can see how from 1968 at around $2, right, $2 per pound, it's gotten as high as $35 per pound, and it's currently probably sitting somewhere between $25 and $35 per pound. And that's the root versus the leaf or herb. So as the price of herbs go up, then you begin to get much more pressure for adulterants to show up. And you have issues around putting leaf material in, right? You can put ground up leaf material in, and it'll have the similar alkaloids, different ratios. They tend to be the opposite. In Golden Seal, you tend to have higher levels of the berberine, hydrastine versus berberine, where in the root it's the opposite of that. And there are different ratios in wild grown versus field grown. So what we have in the isoequivalent alkaloids that are the major constituents of hydrastis is you have these different structures. You have what are called quaternary versus tertiary alkaloids, and that simply refers to the number of double bonds that end up at this nitrogen position. So there are 4 bonds here versus 3. And their chemistry is different, so it makes them difficult to separate. But it turns out there are a number of these isoequivalent alkaloids in a variety of crops. So if we just look at the individual alkaloids here in this north-south versus the plant species at the top, you can see that a number of them contain berberine, all but the Sanguinaria canadensis. So just the presence of berberine isn't enough. You can see that a number of them contain clarithrine. Here's getarizine, palmitine, sanguinarian, and really the indicator of hydrastis is the presence of hydrastinine and hydrastine in combination with the berberine. So this is a work for my own doctoral processes and looking at adulterants, and here we purposely adulterated hydrastis with 5% berberis. Here's your hydrastine, which would have been an indicator of the presence of golden seal, and here's the getarizine that indicates the presence of berberis. So these are ways that on a high end you can account for their adulterations. If you're a smaller lab, how do you do this? This gets to be very difficult. It depends if you have cut and sift material, and you can look at some of the morphology. There is the possibility of using microscopy for looking at some cell components in the root that might allow you to differentiate the fact that you have something other than hydrastis there. It might not tell you what is there, but it would tell you that you have some adulterant species. So these are the kinds of sort of questions and struggles that smaller herb producers have to face is, will I send this out for some sort of higher end methodology that I can't do myself, or will I develop the ability to use a combination of microscopy, and sensorial analysis to ascertain whether my material has adulterants in it. So this is an ongoing conversation for any of you that are really interested in these kinds of questions, particularly if you want to be medicine makers. Then by all means, go to the QA course. It's an intensive, about a week long, and you'll learn a lot of techniques on how to do this on your own. So I want to thank you. This is the program. You can look it up online. There's some real good work going on there, and this continues to be a resource for herbal products manufacturers. Take care until I see you again. I hope all goes well.